Hello, and welcome to my InstrumentL campaign video. My name is Evan Bear, and my research is focused on population genetics of the black-spotted newt, Notophthalmus meridionalis. After watching this short video, I hope you will help my campaign to fund my research. Located in the Tamaulipan Biotic Province, the black-spotted newt is a medium-sized salamander with a very cryptic lifestyle. As an amphibian, the species is important as an indicator of environmental health and can be useful for monitoring human impacts on the environment. Since its description in 1880, the habitat for this newt has been diminishing rapidly. Many historical breeding sites are no longer suitable for the newt populations. This is often due to cattle grazing, fish introductions, and agricultural runoff. For my project, I will be investigating variation among different populations. Right now, I am in the process of finding populations and running traps in nearby locations. However, due to the cryptic nature of the species, just using traps can be inefficient. The species is known to hide in burrows and cracks in the dried soil, some suspect for years at a time, coming out only at night to breed in pools filled after heavy rains. In order to maximize the potential pool of samples, I will be employing a recent technique called environmental DNA, or eDNA for short. The idea is simple. If there is a newt in the water, the DNA may slough off into the water, which we can then collect and analyze. Using a technique called double digest rad sequencing, or dd-rad-seq for short, I will generate a library of genetic sequences. Analysis between individuals will identify unique genotypic markers for each population. With this information, I will be able to generate a map depicting dispersal of the species across the Tamaulipan Biotic Province. Further, this analysis will be able to give a genetic basis for the assertion of subspecies division in the Mexican populations. Here's how you can help. Either by donating or through word of mouth, you give me the opportunity to go out and find more newt locations. With this data, I will be able to create a more detailed map depicting the variation present throughout the newt's range. This data will then in turn be able to be used for future conservation efforts. Now, crowdsourcing for research can sound a little foolish, and understandably, but let's not forget the immortal words of Charles Darwin. I love fool's experiments. I'm always making them. So please, help me in my fool's experiment, and let's save this newt.